In this video, we are going to discuss regarding nitrogen fixation by non-symbiotic free-living blue-green algae or cyanobacteria. In early video, we have discussed regarding symbiotic nitrogen fixation by blue-green algae. Blue-green algae shows a different, they have a different host range in with them they shows a symbiotic association. They fix the nitrogen, atmospheric nitrogen. But now in this video we are going to discuss the non-symbiotic nitrogen fixation by free living blue green algae. Now blue green algae, that is BGA, fix atmospheric nitrogen by two methods, symbiotically and non-symbiotically. We have seen the symbiotic methods of N2 fixation. Now we will study the non-symbiotic N2 fixation by blue-green algae. The large number of cyanobacterial species such as unicellular, colonial, filamentous, filamentous unbranched, filamentous branched or filamentous false branched, heterocystous, non-heterocystous blue-green algae fix nitrogen through non-symbiotic methods. Most of the species belongs to nostochales or stegonematals. The most common examples are given below. First one, filamentous and heterocystous. That examples are nostoc and anamina. Second, non-heterocystous unicellular, for example, Aphanocapsa. Third, filamentous and non-heterocystous, for example, Oscillatoria. Fourth, filamentous and heterocystous form showing false branching. These are example Cytonema and Tolipothrix. And last one, that is non-heterocystous colonial forms, that is Crucoccus. And lastly, that is non-heterocystous form living in bigger colonies. Example is Glyocapsa. Now, mechanism of nitrogen fixation by filamentous and heterocystous blue-green algae is described previously. Only thing is that these members are fixing atmospheric nitrogen by non-symbiotic free-living method. So let's see the mechanism of nitrogen fixation by filamentous but non-heterocystous blue-green algae. Already in previous video we have discussed how heterocystous, the specialized cell of blue-green algae, fix the nitrogen. But here we are going to discuss regarding the blue-green algae who do not have heterocyst and how they fix the nitrogen. So there are different mechanisms of blue-green algae for fixation of nitrogen by free living or non-symbiotic methods are outlined below. See, first method is separation of nitrogen fixation from oxygenic photosynthesis in space. Second one, separation of nitrogen fixation from oxygenic photosynthesis in time. Third one, separation of nitrogen fixation from oxygen is photosynthesis in both space and time. And lastly, switch off and switch on mechanism. See, the first example of this type of cyanobacteria is Oscillatoria. It is very specialized cells instead of heterocyst for fixing nitrogen by non-symbiotic or free-living way. They fix nitrogen in aerobic condition. These blue-green algae members fix nitrogen mainly during night time, in dark period when there is no evolution of oxygen, as photosynthetic activities are stopped which damage the enzyme nitrogenase. So the O2 is also used for respiration, that is dark respiration. It also helps additionally to protect the nitrogen fixing enzymes. So in this way, in dark period when O2 is not evolving and O2 is utilized for respiration, here there is a scarcity of O2 or less amount of O2 is present, and some in this less amount of O2 there the activation of nitrogenase enzyme leads to fixation of nitrogen. This is the first mechanism. The second mechanism in which these species is that there is a temporal separation of nitrogen fixation and photosynthesis. And third mechanism found is that these species, the nitrogenase enzyme is highly tolerant, not damaged easily to the level of oxygen. And the fourth mechanism reported is that these species have specific switch off and on mechanism. Because of these, the nitrogenase enzyme become reversibly active and inactive depending on the concentration level of oxygen. When O2 level is very high, the enzyme become inactive and get protected. But when the level of O2 is reduced, the enzyme become active and perform the function of nitrogen fixation very smoothly by free living or non-symbiotic way. For example, Plectonema. This mechanism play vital role in free living blue-green algae during nitrogen fixation. This mechanism play very vital role in free living blue-green algae during nitrogen fixation. And in short, there is a different mechanism which are listed here, A, B, C, D. Now, here the temporal separation in time means nitrogen fixation takes place only during night, that is dark period, and photosynthesis in daytime, that is light period. Temporal separation in space means the central non-photosynthetic cell engaged in nitrogen fixation while the green cells perform photosynthesis. And however, in some blue-green algae species, both the mechanism are simultaneously work. So these are some mechanisms of nitrogen fixation by filamentous but non-heterocystous blue-green algae, in which the special cell for nitrogen fixation is not present at that time, but temporal separation of space, time, or switch on switch off mechanism are utilized for the nitrogen fixation. Now, like blue-green algae, many free-living bacteria in soil fix atmospheric nitrogen by non-symbiotic methods. For example, Azospirillum, Azotobacter, Rhodopseudomonas, or Clostridium. So these bacteria are associated externally with the host plant or may be completely free living in the soil doing nitrogen fixation, and it benefits associated crop plants. That means when they are present in particular soil and particular crop is taken into that soil, then this soil with this bacteria is having a nitrogen amount which is necessary for that particular plant sown in that soil. So these are associative bacteria, or associative crop get benefits from these bacteria which enhance the soil fertility and which increase the crop plant's yield. And it estimated that the free living bacteria like blue-green algae, these bacteria add 10 to 25 kilogram nitrogen per hectare per year, and these bacteria fix nitrogen without direct interaction with the plant, as they have their own source of energy. This energy they obtain by oxidizing organic compounds. And some free living bacteria have chemolithotrophic capabilities, and hence they can utilize inorganic compounds as a source of energy. Some free living bacteria behave as anaerobes or microaerophiles when fixing nitrogen to protect nitrogenase enzyme from oxygen. And the rate of nitrogen fixation is very low as compared to biological nitrogen fixation. So let's discuss the associative non-symbiotic nitrogen fixation. Different species of Azospirillum are able to form close association with several species of family Poaceae, like means grasses, and agronomically important cereal like rice, wheat, corn, oats, etc. These bacteria fix appreciable amount of nitrogen within the rhizosphere of this host plant by free living methods. That means these bacteria directly do not form any association to the host plant, but they in particular host plant crop farm, they are present and they fix the nitrogen, atmospheric nitrogen, and made available to that plant. That's why they are called as a associative nitrogen fixation by the bacteria. So this is the diagrammatic representation of non-symbiotic nitrogen fixing by free living bacteria, that is azotobacter. So azotobacter are motile, oral, spherical bacteria which are aerobic in nature. They are found mostly in a neutral and weakly basic soil, but not in acidic soil. So let's see what is the mechanism of this bacteria nitrogen fixation in this azotobacter. First, the bacterium has developed a special type of mechanism for protection of nitrogenous enzyme from oxygen. It is a significant intensification of metabolism that reduces the concentration of oxygen in the cell. In addition to this, it has special nitrogenous protective protein to protect the nitrogenous enzyme from oxygen. It has alternative type of nitrogenase. Instead of MOFE nitrogenase, it has vanadium Fe nitrogenase, which is more active than MOFE nitrogenase at a low temperature. It can fix nitrogen at temperature as low as 5 degrees Celsius. Its activity is 10 times more than MOFE nitrogenase enzyme. The nitrogen fixation by azotobacter is an associative type with different types of plant. Many other free-living bacteria like Azospirillum, Klebsiella, Bacillus, Pseudomonas, Azotobacter also fix nitrogen by this method of associative nitrogen fixation without the formation of differentiated or spatial structure. These bacteria to some extent contribute to plant growth also, and hence they are also known as plant growth promoting rhizobacteria. They are called as plant growth promoting rhizobacteria, PGPR. So Azospirillum bacteria are aerobic, chemoautotrophs and gram-negative, and their distribution is extremely wide. The bacteria use malate, pyruvate or succinate as carbon source and hydrogen as energy source. And nitrogen fixation occurs under the condition of microaerobiosis. Klebsiella is more frequently associated with rice, wheat and forage grasses. These bacteria, that is Klebsiella, fix nitrogen under anaerobic condition, and the Bacillus, mainly associated with wheat, is facultative anaerobic free-living bacteria fixing nitrogen. The species of Azotobacter and Azospirillum are fixing significant amount of nitrogen, up to 60 kg nitrogen per hectare per year. So these bacteria are mainly very useful for nitrogen fixation in organic farming type of cultivation, where they fix nitrogen very efficiently because of neutral pH of soil, low level of fixed nitrogen and increased level of carbon. Generally, organic farming practices enhances the diversity and activity of free living bacteria mentioned above. For example, Klebsiella or Azospirillum, or we can say Bacillus, etc. The free living bacteria fixing nitrogen are categorized as 1. Heterotrophic, for example Azotobacter; autotrophic, Thiobacillus; aerobic, Bacillus; photosynthetic, Rhizospirillum. All these bacteria fix nitrogen non-symbiotically in various crops like rice, sugarcane, grasses, etc. Now the ammonia or fixed nitrogen by these non-associative nitrogen fixers is assimilated in plants. How? The free living bacteria fix nitrogen non-symbiotically as described before and convert nitrogen to ammonia. This ammonia is assimilated by the plant into different compounds they require through the involvement of three main enzymes which are main route of ammonia assimilation. First enzyme, that is GS, glutamine synthetase. Second, GOGAT, that is NADPH dependent glutamate synthetase. And thirdly, GDH, that is NADPH dependent glutamate hydrogenase. However, GS is the key enzyme or route for ammonia assimilation. So this is all about non-symbiotic nitrogen fixation.